So it's my pleasure to open this seminar. There are not so many people in the room, but a lot of people are connected. And it's a special seminar because we will have the general lecture as a part of the process for professor appointment for docent or associate professor Martin Reichard. And to become a professor is the big thing, and it's also a long process to go. We are still at the beginning. Maybe after this talk we will be halfway, and so this talk will be evaluated by the committee, and the committee will also evaluate scientific and teaching performance or achievements of the candidate. And there are 5 members of the committee. One has to be from abroad, and it's Professor Christina Seth from the University of Greifswald, Austria. Hello, Christina. Hope you can see us. And the next one is Professor Petr Raab from the Academy of Sciences of the Czech Republic. Hello. Then the third one is Professor Zdeněk Laštůvka from Mendel University here in Brno. The fourth one is Professor Miloš Macholán from our department and also Academy of Sciences is here. Welcome. And myself as the chair of the committee. So once we agree that the quality of the candidate is enough to go through the process, and I guess it will be a really difficult task for us to judge. Then there are another two talks that needs to be given, and the first one is in front of faculty council, the voting, and another one in front of the university council, and also voting. And if you think that it's over, not quite yet, then all the documents have to be sent to the Prague, to Castle, by hands of our beloved president. And from the media we all knew what's the common practice there. But I have high hopes, because most of the Martin work is dealing with fish, and fish means water, and water means the channel. So I'm very positive about this. Okay, so now I should briefly introduce the candidate. He graduates here at our university, and that time it was the Institute of Systematic Zoology and Ecology, I think, which was one of the 2 nuclei of our current Department of Botany and Zoology. Then he spent some time in abroad for a couple postdoc positions, and then got a permanent position here at the Institute of Vertebrate Biology, Academy of Sciences. And recently he also obtained a part-time job at our department. Most of his work deals with fish ecology, fish evolution, evolution of parasite, host-parasite interactions, mechanism of aging. He'll definitely tell us more about this, all those topics. And he's a very fertile scientist. He published more than 140 scientific papers in mostly, or in all cases, in well-respected journals. Among those are journals like Cell or Molecular Biology and eLife, so really top-ranking journals. And the quality of works also documents well number of citations based on Web of Science database of Scopus, which reached almost 3,000 references, and therefore also his age index is very high. I think it's 38 right now. And for the younger researchers and students, it means that 38 of his papers have been cited 38 times each. So it means that many of them have been cited much more than 38. And he's also active in public outreach activities, and One for All. Part of his research, or results of his research, also find a way to one of the last David Attenborough movie series, which named 7 Worlds, 1 Planet. And it appeared in a part devoted to Africa. And if you are shooting about Africa, it's, you know, there are a couple mandatory things you have to show people. Zebras, elephants, you know, the gorillas, a chimp has to crack a nut with a stone, and then all of a sudden, you know, Martin's cuckoo catfish. So it blows your mind. And okay, now I have to leave you on this very high note and let Martin talk. So it's your turn.
Thank you very much.
Remove your mask.
Thank you very much, Michal, for kind introduction. Michal, Miloš, other members of my evaluation committee, dear colleagues and friends, thank you very much for finding time to connect online to watch my presentation. I tried my best to accommodate slides to this very special occasions where you will watch my talk on your computer screens. So I try to minimize any bright colors and shining letters. The title of my talk that has been chosen by my evaluation committee is Host-Parasite Relationships in a Dynamic World. And I think that this topic deserves some clarification. This is because parasitism is extremely widespread life history strategy. And we tend to think of parasites as some tiny creatures that extract blood or some other resources from bodies of much larger hosts. But this is not the whole picture, because many parasites can actually be larger than their hosts, and they may extract other valuable resources. For example, parental care is very expensive and very valuable resource, and there is a group of parasites termed brood parasites who relinquish all their parental duties onto individuals others than closely related to them. So when we think or when we speak of brood parasitism, we mean biological parents that pass all their parental duties onto other individuals. This is a very common strategy within species, but this is not the type of the strategy that I'm going to speak about today. Instead, I would like to concentrate on occasions of brood parasitism between species. So it means that parents of one biological species pass all their parental duties onto individuals from a different species. This life history strategy has evolved, for example, in birds independently at least 7 times. And I think you are all familiar with cuckoos, but there are also cowbirds, honey guides. There is even one species of brood parasitic duck. The same set of brood parasitic strategies have evolved in social insects. There it's typically termed social parasitism, but from evolutionary point of view, it's the same strategy. So there are brood parasitic wasps, ants, bees, or ambrosia beetles. Now, there is one problem with this definition, and this is especially more pronounced in Czech language, because when you speak of brood parasites, you have a tendency to think about situations when the parental species and the foster parents are quite related. So, for example, all avian brood parasites parasitize parental effort of some other birds. But there are many more cases of reproductive parasitism. So let me to define this term more broadly as reproductive parasitism. If you concentrate on those 3 photographs on your left side, they describe the life cycle of one group of butterflies. In English, for example, alcon blue or large blue. And those parasites evolved a strategy that includes parasitism to other insect groups. So the first few instars of of caterpillars of this butterfly feeds on host plant, but then they drop down on the ground and they try to mimic pheromones of Myrmica ants. And if they succeed, then passing Myrmica ants, believing that they found a straight offspring of their own, carry those caterpillars into their nests, where they nourish them for many months, and at the end of the winter, or in actually late spring, early summer, those butterflies emerge from ant nests. If you define reproductive parasitism such broadly, then you can also include cases like parasitoid wasps that lay their eggs inside or onto bodies of other insects. Now, as Michal explained, I am primarily a fish biologist, and there are many examples of reproductive parasitism among different fish species. So, for example, this picture in the middle, the blue one, shows a snailfish, a marine fish, white species rich lineage, that is a parasite of marine crabs. They lay the eggs on their, inside their gill cavity. The same or similar thing is also typical for bitterling that parasitize freshwater mussels. And this is one model system that I will address in more detail later in my talk. There are also some true brood parasites among fishes. There is actually only one confirmed case of obligatory brood parasites. So obligatory means that this is the only way how they can reproduce. And this is cuckoo catfish parasitizing African mouse-brooding cichlid fishes. And I will speak about this system also later in my talk. What you see on the top is a cyprinid species, a minnow from Japan, that has evolved facultative brood parasitic strategy. So when they can, they lay their eggs into the nest of some other fish species, but if this is not available, they would lay them elsewhere. Now, what I mean by dynamic board. So let me define dynamic board at 3 different levels. And the first one, this is really the level of a long-term coevolutionary dynamic. At the second level, I will speak about ecological time, and by ecological time, I mean a possibility to see very fine scale coevolutionary dynamic, for example, through some disturbance to an ecosystem or to a biological community. And the third layer of this temporal scale is individual lifespan. So within individual of a single host or a single parasite, there could be acquired immunity after repeated infections. Hosts can be better able to defend to the parasite. They can also learn how to avoid it or tolerate. So coevolution is reciprocal, or is described as reciprocal changes between 2 co-evolving partners. And let's define those partners as species for the scope of the talk today. This is not necessarily the host and parasite. It could be predator and its prey. And Michal wanted to see some zebra, so I have zebra here as well. It could be pollination, seed dispersal, and other ecological strategies. We can describe coevolution as a dynamic process. So at one end of continuum, we can see coevolutionary arms races. So they involve cycles of adaptations and counter-adaptations, and we tend to associate them especially with negative interactions. However, there is also a second end of this continuum, and this is evolution towards some equilibrium state, where both partners are more or less happy in the relationship. And again, we tend to associate them primarily with positive interactions, like with symbiosis, but we should also include a host-parasite relationship, because some hosts tend to tolerate the parasites. Now, if you study coevolution, you can look at large-scale coevolutionary patterns, co-speciation, co-diversification. But there is also alternative approach. And this is the approach that I take in my research. And this is to look at co-evolutionary processes. It is the dynamic nature of coevolution. So it means that you study adaptations and counter-adaptations, and ultimately, of course, those changes are genetic, but you can observe them at the level of physiology, immunity, or individual behavior. So the second temporal level is ecological time, and I said that you can look at them very effectively through some, let's say, human-altered changes to ecosystem, or inclusion of new species to biological communities. Well, the best known and perhaps textbook example of changes within ecological time frame is the case of the peppered moth, a common moth species in Europe. What you can see on the screen are actually two individuals of the moth. The one on your right side is the most typical form, light form, and if you can't see it, it means that it is because it's cryptic, and it's cryptic because it needs to be safe from visually hunting insectivorous birds. Then there is a second form, extremely rare under normal circumstances, melanistic black form. What has happened in Britain about 2 centuries ago during Industrial Revolution with a lot of air pollution, then there was suddenly a lot of much darker surfaces to sit on. And within extremely short time frame of very few generations, there was a rapid shift into increased proportion of this black moth. Another possibility to look at ecological time frame is to study cases of novel species and communities. We tend to think of invasive species as those that can alter entire ecosystems, like zebra mussel in American Great Lakes or Nile perch in Lake Victoria, where it caused extinction of many local endemic fish species. However, in most cases, the impact of non-native species on natural communities is much more subtle. So let me show you the example of cane toad invading in Australia. Cane toad is originally South American amphibian that has been introduced to many parts of the world, including Australia. And what happened in Australia was that many of the native species adopted this potential prey into its diet. So what happened was that many of those predators suffered very severe mortality because toads are toxic. And this toxin was not found in Australia before. So the predators were evolutionarily naive to this type of prey. However, there was a very strong selection, for example, in at least two snake species, towards much smaller gape size, because those individuals who had heads small enough not to swallow very large toads survived. So within very few generations there was this morphological adaptation. Obviously, there were other forms of adapting to this novel prey, and those individuals who attempted to feed on toxic toad and survived that attack, they very quickly learned aversion against this particular prey item. So this leads me to the third temporal level, to individual lifespan. And you can imagine that it's really mounting immune response following repeated infections. But as I will try to emphasize later in my talk, very often it can be also learning to either avoid or tolerate a parasite, and also learning from the side of the parasite, perhaps to better trick the host next time. So after this conceptual introduction, I would like to follow with focusing on the research that I have done in the past, and I will specifically concentrate on two model systems. The first one is interaction between betterling fishes and freshwater mussels, and the second is relationship between cuckoo catfish and Lake Tanganyika cichlid fishes. Now, I have been advised that during this talk today, I should highlight some significant achievements that I have made in my career. And if I should name one, it's certainly that I had opportunity to work with this group of very talented and very dedicated people. So those are members of our team at the Institute of Vertebrate Biology here in Brno. And it was really a pleasure to work with them over the years. And you can see current or the most recent members, but there were many others who worked with us. So when I will speak about particular case studies, please keep in mind that they have been accomplished by cooperation within our large group. So let's start with the betterling system. This is male betterling, and during reproductive season, male betterlings defend small territories near freshwater mussels. This is female betterling, and I hope that you can see extended ovipositor that she uses to lay her eggs into the gills of freshwater mussels. Then betterling eggs and embryos develop for a couple of weeks, for about 1 month, and I hope that you will be able to see betterling spawning. This is a female betterling inserting her ovipositor, and male betterling releasing sperm. This will be repeated again. So female quickly inserting her ovipositor and then male. Now, this video has been slowed down by the factor of 6. So in real time, this action is much quicker. What happens in the gills of betterling is development of embryos until they are able to actively depart the mussel as fully developed juveniles. Now, this is just one part of this interesting story. The second part involves life cycle of unionic mussels. So, unionids have internal fertilizations, and then they develop larvae termed glochidia within the gills of maternal mussel. So, potentially, betterling and mussel embryos and larvae can meet and overlap spatially and temporally in the gills. When glochidia are ripe, they are released from maternal mussel, and within few hours or few minutes even, they have to attach to a fish host. It can be typically fish fin, it can be gills or some other parts of the body. But because hosting glochidia is very costly for fish, very often those glochidia are not successful and are rejected by the fish. And the fish can be betterling or some other species. So there is a range of potential costs and benefits. Most important are for the betterling that they have safe place for incubating their eggs, and potential costs that they are more susceptible to glochidia because they spend a significant amount of time near mussels. For mussels, They have easily accessible hosts for the Glochidia, but at the same time, beetling embryos in their gills may be actually quite costly. So, then what you can see is a range of potential relationships from mutually beneficial to reciprocal parasitism. Now, beetling are East Asian group of fish. There is a listed species in East Asia, in China, Korea, and Japan mainly, and there is one species or species complex in West Palearctic. All beetling oviposit in freshwater mussels, and in East Asia, very often several, I mean 8 beetling species may coexist locally. And if they do so, they very often have access to multiple mussel species. So there is a diversity of beetling, and also there is much higher diversity of freshwater mussels in East Asia. Here you can see diversity in the size and shape of their shells, but of course they are also very diverse in their internal structure. So, as I said, one possibility would be to look at coevolutionary patterns. I'm not going to do that, but I would like to highlight a few points. What you can see now on the screen are different types of beetling ovipositors. They are very different functionally, and they are adapted to be used for oviposition into particular mussel species. What you see here is a selection of the diversity of beetling eggs. Again, they match corresponding gill structure because they have to wedge themselves inside the water tubes of mussel gills. And this is a selection of beetling embryos. Again, they are still faced with necessity to anchor themselves in the mussel gills. So, as you would expect from most parasite lineages, there are some generalist and some specialist species, and their traits are adapted to particular type of mussel gills. So what we believe is that actually there is coevolutionarily driven ecological speciation in this coevolutionary relationship between beetling and mussels. Now, one of the highlights of my research in distant places is that I really try to blend and disappear in local culture. And there is one other point why I use this slide, and typically I use it to introduce my collaborator, Professor Liu Huanzhang, saying that he's from the Institute of Hydrobiology in Wuhan. And until this year, I used to tell, well, this is a provincial city in central China that nobody in Europe knows. But obviously, this has changed a lot over recent months. And I also had to cancel my trip to Wuhan in March this year. So let's have a look at the coevolutionary dynamic between beetling and mussels. We can visualize any coevolutionary process, not necessarily between host and parasites, and I have adopted, not knowing that Michal will introduce it this way, a relationship between predator, lion, and prey, zebra. And you can follow this relationship between any two co-evolving partners through evolutionary time. And at certain period of time, one of the partners can have the upper hand, but in some other time period, there could be adaptation of the other partner to escape from this relationship or to have the upper hand. The problem is that we cannot study those long-term temporal changes in real time, because this is a time span of thousands and thousands of years. But there is a solution to that, and that solution is then we can look at the spatial mosaic of regions where the two species co-evolve, because they can co-evolve for a different length of time with a different strength, or maybe following a different temporal dynamic. Of course, this is even more complicated because they are not isolated from other part of the biological community. But the point I want to make is that we are able to look at a spatial mosaic, geographical mosaic of coevolutionary states. So if you look at European beetling, it is the parasite of Unionic mussels in Central Europe. And we know it because we have found that hosting beetling embryos is costly for mussels. They stop growing, and this means that they have much lower reproductive output. And at the same time, beetling are capable to avoid parasitism of Glochidia. However, this is situation in Central Europe, and beetling has very wide range throughout Europe, and the same is true for its host mussels. So we wanted to know whether the same relationship or the same state applies, for example, for other parts of their sympatry, like Southern Europe. So we know that in Central Europe, beetling is relatively recent, while in Southern Europe, the sympatry between freshwater mussels, and these are the very same species of freshwater mussels than in Central Europe, is much more ancient, much older, perhaps about 2 million years. So when we tested this, we found that even in Mediterranean region, in Turkey, beetling are parasites of freshwater mussels, but they face much stronger responses. So many more eggs are ejected by mussels, and this is perhaps because mussels can close their siphon much more rapidly. Also, some eggs that are inserted into the gills are not properly placed and don't develop. Okay, so this was a natural contrast between two regions. But what is happening at increasing rate is that different species are transported from their original distribution, from their original range, to new parts of the world. And I have taken an example of this nematode parasite, Umbilicoides crassus, which parasitize naturally Japanese species of eels. What has happened was that those Japanese eels were imported to Europe for aquaculture, and this aquaculture attempt was not successful, but what was successful was the introduction of this parasite. So they started to exploit coevolutionarily naive European eel, and it caused massive mortalities. This is because they were at a different phase of this coevolutionary arms race. Obviously, there are many practical consequences that, especially nowadays, we are all aware of. But there is one other point that I want to make. And this is the fact that we are typically concerned with the effects on hosts. So we speak of parasite spillback when a native parasite can reproduce in a non-native host and then spills back into the population of native hosts, or we speak on parasite spillover when non-native species bring its parasite fauna and it can start infesting native hosts. Well, what I would like to say that we should be also fair to the parasites and try to look at this coevolutionary relationship from their perspective. And to do so, of course, I will use my favorite parasite, which is beetling fish. And as I said, in Europe, beetling can parasitize 4 vitally occurring European mussels, 2 species of Anodonta, 2 species of Unio. But recently, a few decades ago, there was invasion of non-native mussel from East Asia. So this is the mussel from the region with quite high bettling diversity. So when this happened, we asked 2 main questions. The first one was whether Glochidia of this invasive species can also parasitize bettling, and what is the response of the bettling to this new potential host. So conceptually, what we see here is translocation from region with very high bettling density and diversity to the region where selection on those parasites was extremely low for maybe 2 million years. So first, we looked at glochidial infections, and most of the work that we do on glochidia or on freshwater mussels in general are done with collaboration with Karel Douda from Czech University of Life Sciences in Prague. What you see here on the screen is the fact that for a control European species, we confirmed something that we knew already, and this is that European mussels, glochidia of European mussels, cannot develop well on the bettling. What you see in those red columns is the fact that this invasive mussel, Anodontis woodiana, is perfectly capable to exploit bettling. And is actually capable to exploit any other freshwater species that we have tested so far. So this is one part of the story. The second part is whether bettling can use this mussel as a new host. So we designed an experiment in central Poland when this invasion proceeded for quite a long time. And we, after 3 weeks of exposure of a set of native mussels and this non-native one, we look at the number of bettling eggs and embryos on their gills. What we found was very striking and very clear. There were many bettling eggs and embryos on European hosts, but there was not a single bettling embryo in the gills of woodiana. So you may think that this is the end of the story, it's just not a good host. But the problem is more complicated. And this is because over those 3 weeks, we also observed the behavior of individual bettling pairs towards different mussel species. And we have found that woodiana is actually used quite often for oviposition. What is happening is that most of the eggs are immediately ejected. Within 1 or 2 seconds, the eggs are ejected from mussel gills. And those ejections continue, and after a couple of days, there is not a single bettling embryo in woodiana mussel. So what we see here is the fact that European bettling is unable to exploit this new mussel, and at the same time, bettling is being parasitized by this mussel species. So effectively, what we see here is complete reversal in the relationship between the host and the parasite. So the former parasite became a host, and the former host became a parasite. Well, the story gets even more complicated. And this is because when we started to find woodiana also in southern Moravia, in the river Kiovka, we wanted to continue this research, and we wanted to replicate some results. And what we have found for those woodiana from the Czech Republic is that they actually were not chosen by the bettling for oviposition. So they considered them as a potential host because they inspected them, but they never make decision to lay their eggs inside. So we thought that maybe we observed a range, a shift across coevolutionary mosaic. So multiple introductions maybe different populations of Woodiana were introduced to different parts of Europe, and that's why we see this contrasting response. To confirm that this is the case, we teamed up with Adam Konečný here from the Department of Botany and Zoology, and he modeled genetic data. He modeled invasion history using genetic data. Well, to our surprise, he found that there was only a single colonization event. So it was from the Middle Yangtze River Basin. But initially, those invasive populations were isolated in two parts of Europe, in southern France and in northern Romania and southern Hungary. But after that, there were repeated waves of admixture because those mussels, as Lochidia, parasitize any freshwater fish, and those fish are used for commercial transport for aquaculture and other purposes. So irrespective of the enigmatic reason why those two populations behave in a different way, we wanted to know what is the real impact on European bettling populations. So we designed an experiment that should look at the population dynamic, or a population consequence of this invasion. So we had three treatments. The first one was with native mussels, and then the other two had population of native mussels invaded by either this Polish population of Woodiana that can eject bettling eggs, and then the other one that is not used. There's one other point, and this is that mortality of bettling embryos inside the gills of the mussels is density dependent. So even if this new host is not used, but replaces some of the individuals of native mussel species, it still can be detrimental because it decreases bettling embryo mortality due to density dependent process. And as you may expect, we have found that indeed the biggest impact and the significant impact was for those mussel communities with a population of Woodiana that was capable to eject bettling eggs. So there was significant decrease in the number of juvenile bettling departing from the mussel. So, well, this experiment was actually even more complicated because we used multiple bettling populations, but I'm not going into that detail. But the main message from this experiment is that there are population-specific effects and consequences, and that this is all driven by behavioral discrimination. Now, there are more troubles for bettling, and the new potential trouble is this East Asian species of bettling that is capable to survive in Central Europe, and it has already become invasive in some other parts of the world. It's now available in aquarium trade, in pet shops, but as far as we know, it still hasn't been recorded from the wild in Europe. The important point is that this species of bettling is perfectly capable to use this invasive mussel, Synodonta woodiana. So we first look how the two species, well, using experimental approach, of course, in captivity, we looked how they would compete for mussels. And what you see here is that European bettling is much more aggressive and is capable to displace Asian bettling from mussel hosts. This is also mirrored in the number of eggs laid into mussels, so there is about 3 times higher number of eggs laid by European bettling. But remember one thing, and this is that this Asian species of bettling can use Asian woodiana, while European bettling can't. So to look at the population consequences, we designed 2 mussel treatments. The one had a community of native mussels, European mussels, and the second treatment was invaded by woodiana. And we contrasted them with 3 fish treatments. 2 of them were controls, so either European or this East Asian bitterling alone, and the real treatment was the mixed community of the two species. When you look at the controls at experimental populations with single bitterling species, you see that irrespective of the mussel community, there is no difference in their reproductive success. However, when they have to compete, there is a large advantage of the native European bitterling when they coexist with native mussels. However, this advantage disappears completely when they are housed together with this invasive mussel species. So the main message here is that the actual fate of potential invasion, and remember that this invasion is really potential, it hasn't happened yet, is dependent on the success of establishment of this non-native Woodiana mussel. Now I think I'm running short of time, so I will very quickly pass through this third layer, disease ecology, and perhaps one study I will choose to detail a bit is the fact that we know that bitterling are resistant to glochidial infection. And one possibility could be that they acquire immunity during their embryonic phase, because they reside in mussels and they are in close contact with them. So to do so, we produced some in vitro bitterling that has been never in contact with mussel tissue, and we actually found that they also were not parasitized. So perhaps this immunity is innate. We also looked at the energetic cost of hosting bitterling for mussels and hosting glochidia. And we also look at some behavioral response to repeated exposure to glochidia, but I'm going to skip that now. So let me to summarize the lessons we can take from bitterling mussel system. First, I believe that bitterling adaptations to coexistence with mussels can lead to ecological speciation. The second is that we can observe coevolutionary dynamics at a very fine scale. And the third point is that I believe that actually invasive species can be used to illuminate some interesting evolutionary questions. So let's move on to the other system that I use in my research. And this is Cuckoo Catfish parasitizing mouse brooding parental effort of Lake Tanganyika African cichlids. Mouse brooding is a parental strategy that has evolved several times among fishes, but it's best studied in cichlids. Cuckoo Catfish parasitize this effort. Okay, so I just here have a short clip. So what you see here is female of Lake Tanganyika cichlid, and this is an example of mouse brooder. So she collects the eggs that are freshly laid to take care of them in her buccal cavity, in her mouth. This is male cichlid, and he flushes his anal fin to discharge sperm and to encourage female to spawn and to fertilize the eggs. So we see this is female laying her eggs, and they are taken into her mouth. This parental strategy, and then she would care for those eggs for the period of at least 3 weeks. This parental investment is parasitized by cuckoo catfish. Cuckoo catfish are one of the small species flock of cynodontist catfishes in Lake Tanganyika. There is about 11 species, but only one, as far as it's confirmed, is parasitic. And they are quite common in the lake, and they predate actually on eggs of spawning cichlids. So what you see here is predation of cichlid eggs by cuckoo catfish, but during that they also spawn their own eggs. And as female cichlid panics and starts to collect her own eggs before they are predated, she also, by mistake, collects some of the eggs of the cuckoo catfish. So what happens then is that she takes care of her offspring for a period of those 3 weeks, which is very, very costly. This strategy has been described many years ago, but there was basically no research on this topic. Until about 2 years ago, when our research group and also one American group started to publish first studies. So what we know at the moment about the system? That American group, now led by Marcus Cohen from Harvard, they described embryonic development of the cuckoo catfish and their cichlid hosts. So as you can see, the eggs of cuckoo catfish are really not mimetic, they are much smaller and different shape, but they develop much faster. Cuckoo catfish hatch very quickly, and since the age of 4 or 5 days after fertilization, they start to feed on host progeny. They have very good adaptations to do that. So, for example, at the age of 6 days, what you see here is a comparison of the cuckoo catfish embryo and the embryo of closely related Cynodontis species from Lake Tanganyika. So they have much larger mouth, and they also have very well developed embryonic dentition. So what happens over that period of 3 weeks is that the host brood is typically completely eliminated, and sometimes even cuckoo catfish start to cannibalize on each other. One interesting question about cuckoo catfish parasitism is its origin, because in all other cases of brood parasitism, it has evolved from parental care. This is very different in Cynodontis, cuckoo catfish, because ancestors of this species had no parental care at all. I think it's a very interesting question, and this slide perhaps shows how small is the world of cuckoo catfish research, because there are two hypotheses that try to explain the origin of this brood parasitic strategy, and they were both formulated by members of our team. So the first hypothesis believes or suggests that brood parasitic strategy started by exploitation of very strong maternal instinct of cichlid mothers, because they tend to collect any stray offspring. So in a situation like this, a mouse-brooding female can actually adopt freely moving catfish embryos. The other hypothesis that I have formulated believes or suggests that brood parasitism has been preceded by predation. So first, cuckoo catfish was a predator who specialized on spawning cichlids, but since spawning in fish involves a lot of hormonal signaling that is transmitted through aquatic environment, maybe they have started to be affected, and they started to reproduce during this predation. We now have made a mathematical model that actually shows that predation greatly facilitates the evolution or the origin of brood parasitism. One other point I want to make today is that I strongly believe that if you do experimental work, it should be very solidly rooted into the knowledge of natural history of your target species. So I really try to combine experimental work with field studies. And we were lucky enough that we managed to organize an expedition to Zambia last year. And during a couple of weeks, we have collected almost 800 mouse-brooding females of many different species, and some of them were parasitized. Now, I don't want to devote much time to this field study because last week there was a seminar by Radim Blažek at Parasitology department, and it would overlap, and I think all audience overlaps to a certain extent. But I would like to highlight a few points. The first one is that we believe that what we may see in cuckoo catfish are host-specific lineages, and we have already some evidence that there are at least two already described species being brood parasites, Cynodontis multipunctatus and Cynodontis grandiops. And now we have genetic data from naturally collected broods, and we can test whether they use different hosts or not. We also did the research not only on the host cichlid species, but we have also get a lot of information about cichlids that are potentially hosts, but they are not used by the cuckoo catfish. We are interested in the mating system because they spawn in a larger group, and we also collected some samples that we would like to use to understand what makes cuckoo catfish so special. Instead, I will very briefly introduce our experimental work on this form of parasitism. And in the study that I will devote most of my time now, we contrasted, or combined, not contrasted, but combined coevolutionary history of the host, so hosts that are sympatric or allopatric with a parasite, with individual experience, whether they have been exposed to that parasite or not. And this study had two phases. The first one was largely descriptive, and we found that cichlid species from Lake Tanganyika, so those that are coevolutionarily experienced with cuckoo catfish, they are parasitized at much lower rate. In contrast, most brooding cichlids from other African lakes are parasitized to much higher extent. But the second part of that study is, I think, much more interesting. There we adopted experimental parasitism approach, and we produced in vitro eggs of the cuckoo catfish, and we then inserted them into the buccal cavity of recently spawned female fish. We then isolated that female, and we checked and inspected her tongue for any ejected offspring, parasitic or her own. And we also looked at the survival of this brood over the whole period of brooding. So what we have found was a very striking contrast in ability to eject parasitic eggs between sympatric host, so it was over 80%, and allopatric, so evolutionarily naive host. It was very few, 10–15%. There was no difference between individually experienced and naive females. But this came with a cost, because those sympatric hosts, they tended to eject also some of their own offspring, perhaps in an approach to get rid of the parasitic egg. But this has not happened in allopatric hosts. So for the survival of the parasite over this whole period of 3 weeks, this is a corresponding value. Again, there was no difference between individually naive and experienced females, but survival of parasitic offspring in evolutionarily experienced host was very low, in contrast to survival in allopatric host. So this is the same figure shown here for comparison, because what you see on this other graph is survival of host-own broods. In black bars, this is when they were not parasitized, so we handled them in the very same way, but they just received a stream of water instead of the eggs. And the red columns describe survival of host broods when they were parasitized. What is striking here, that the only combination that was successful in mitigating potential cost of parasitism were individually experienced females of this sympatric species. So what I think we have seen here was first the fact that sympatric hosts are much better in avoiding being parasitized by cuckoo catfish, but also these evolutionary experienced females can learn and mitigate potential cost of parasitism. So the last experiment that I'm going to present today is looking at this system from the perspective of the parasite. The parasite, cuckoo catfish, can potentially also learn over its lifespan how to parasitize their hosts. So what we have done was that we had 3 groups of cuckoo catfish. The first one was completely naive, so they were produced by in vitro fertilization and they have never seen a cichlid host. The second group were of matching age, but they coexisted with host fish for several months. And the third group were something that we called super experienced. These were fish that we have in the lab for many years. So they really were experienced in parasitizing their cichlid hosts. And even within those 3 groups, we were interested in temporal pattern. So at the beginning, maybe they were not very successful, but they may learn over the period of a few weeks. So we divided the duration of the experiment into 3 periods. And what you see here is that indeed, individually naive parasites, cuckoo catfish, they were much less capable to successfully parasitize their hosts, but they learned very quickly and they were on the trajectory to match the experienced parasite within the framework of a couple of weeks. Now, those cuckoo catfish, they are very long-lived. They may live for 20 years, maybe even a bit longer. There were also consequences for the hosts, because those more experienced parasites, remember that they are also predators of host eggs, they have eaten many more eggs of their hosts, even in those spawnings that were not parasitized. So the lessons we can take from the cuckoo catfish system is that I believe that they are a powerful system to understand the origin of brood parasitism, but we don't know the answer yet. And they are also quite convenient experimental models to test different coevolutionary questions. So let me just with this last slide conclude the most important points that I wanted to make today. The first one is that host-parasite relationships are extremely well suited to understand coevolutionary processes. The second is that we should broaden our interest and move from solely host perspective to parasite perspective, so to look at these associations from the perspective of parasites. The third point is that I believe that human-induced changes are very good, well suited to test some interesting evolutionary questions. And the last point is that hosts and parasites keep contesting across various timescales, and this is a struggle that never ends. With that, I would like to thank you for your attention, and I hope that we have time for discussion. Thank you.